Hello everyone, in this tutorial we are going to talk about the inducible transgene expression and what this is all about. So most of the time, once you have your transgenic mice, it usually carries a gene that may lead to the expression of a condition that may become lethal, such as a disease, a cancer type, something that may kill the mice as it starts expressing that specific gene. And that is something that we do not want in a research. So one of the way that we do it is that we try to control. So we will try to control the expression of that potentially lethal gene in a specific body tissue or organ and at a specific time. A good example is you want to study brain cancer and you have a mice that has a transgene that can express brain cancer. But the problem is that if you don't control its expression, from the moment the embryo fertilizes and it's growing, the mice is going to have cancer. And by the time that mice is born, it's going to die of cancer and you won't perform your study. So how do we control the full gene in such a way that it only gets expressed at a specific time? Because remember, there's a difference between carrying a gene and expressing the gene. So to overcome this, scientists uses the system known as, or a strategy known as the TET-off system, where your transgenic expression would depend on the action of an inducible transcriptional activator. Let's look at this figure A, for instance. This transcriptional activator, by the way, is controlled by the level of an antibiotic such as tetracycline. That's the reason why it's known as the TET-off. So you are going to have a promoter region and you are going to have your tetracycline transactivator, which we are going to refer to here as TET-A. In order for us to have this green, which is a gene expression, in order us to have a gene expression, you want your TET-A to bind to a promoter. Just if you're very confused, just think about the lac operon. There was the repressor and operator. So it's more or less, it's not the same, but there are certain similarities there. So your TET-A binds to your TET-op promoter. As soon as these two are bound, RNA polymerase can sit there and your protein of interest is going to express and you have your desired phenotype or physiological reaction. Understand? Now, this works well in the absence of tetracycline, your antibiotic. Okay, I'm just going to put tetracycline. When your tetracycline is present, this tetracycline, your antibiotic is present, what is going to happen is that the antibiotic is going to bind here at a region under this transcriptional activator TET-A. If your antibiotic is bound here, which means that this TET-A will not sit on your promoter, and as a result, transcription is blocked and you are going to have no gene expression. Understand? So how do we actually apply this in our research? So the way this works in our research is going to be when you create your transgenic mice, what you would want to do is you would want to have one mice that is carrying your TET-A and the other mice is going to carry your TET-op in this following manner. So what you're going to have is you would have one mice here, I'll say in blue. As you see, it's carrying your TET-A, and since we are trying to study here are brain tumor. So the mice that carries the TET A, now you know this is the TET A that binds to the TET up. It's going to carry this TET A only in the brain, in this organ. It's only in this one tissue. And you're going to have another transgenic mice here that is going to carry your TET up X. So this bottom part here. So one mice is carrying this. I'm going to put this here, mouse 1, and your second mouse is carrying this. And once you have this, there's 2, and the mice that carries your TET up obviously is also carrying your gene X, your gene of interest, my gene X. And once you have these 2 mice, you are going to crossbreed them, and they're going to give you an offspring. This offspring that you see is going to carry both the TET up and also your TET A. Both of them are carrying this TET A and your TET up. Why? Because it's a crossbreed, it's an offspring of these 2 mice. Now, what you're going to do in order for you to control the expression in the presence now, where when you have tetracycline, let's say in this mice, if you keep feeding it tetracycline, you are going to have the scenario that we had here in front of us. This scenario here, you're going to have more or less no expression. Let me clean this one. Now that you understand it a little bit, let's go back. So in the presence of tetracycline, this is what's going to happen. This scenario here is going to happen in the presence of tetracycline. The tetracycline is going to block TET A from TET up, and as a result, you would have no gene expression. All right. And as soon as tetracycline is absent, the second outcome is going to be now this one. In the absence of tetracycline, you are going to have this reaction here. TET A is going to bind to TET up, and you're going to express your protein. And since you carry TET A only on the brain, then this mouse will only express the gene on the brain. As a result, it's going to have a brain tumor. So that is how your TET of system work. Feel free to stop the video, pause, watch it as many times as you want to until you understand it and it's all clear. Thank you so much.